Now we have Dr. Albert Gideon from Berlin talking now more about bringing in genetic tools to study the brain, and looking forward to your talk.
Thank you. It's okay. It's probably better to point with the mouse, right? Because, oh. So if I do this, it doesn't work. And if I do, keep that up. So in the meantime, so it's an honor to be here. Thank you very much for the invitation, Taufik and the Toronto gang. It's been a pleasure. Yeah. I'd like to share with you today some unpublished results. That, by the way, this is, I understand this is last session after lunch, so you're all kind of tired. I'll try to talk slowly and to finish quickly. So, there we go. Perfect, thank you. And the guys with the, yeah, 5 minutes? Okay, thank you. So I'll share with you a set of unpublished results. Nothing that I'm showing you now has been published. And we'll start with some of the motivation. So I'm going to cite Christopher here, who is citing Ed Boyden, who probably cites somebody else. And the point is that, I mean, it's obvious that one of the important things that we want to do in the brain is to kind of map the connectivity and understand the function that kind of underlies this connectivity. So I don't think I need to say a lot about this. And the goal that we kind of put to ourselves is after, so let's say it like this. We want to understand the human functional brain microcircuit because it's a fundamental aspect of understanding the human brain. This is something that we cannot get away from. And there is a barrier for doing so, and that is the important thing. So some of the studies are just excellent and very difficult to do, like we heard in the last 2 days. There are some limitations, and they are not available to every laboratory. So, and of course, what we heard today, working in cultures is excellent in so many dimensions. And we want to, when we work on the microcircuit, our goal would be to keep it as fresh as possible. So you can learn a lot from the cultures, from the slice cultures, but if we had the access to the fresh slice to do the physiology and circuit mapping, that would be just fantastic. And this is the kind of experiment that we are after, right? We want to do the same thing that we do in animals, like we inject the virus, and then we slice the brain after some time, and then we do our experiments, right? Why don't we have that in humans, right? And I'll show you an example of experiment that is done, everyday kind of experiment in our laboratory, which is a circuit mapping using Campari, which is a ratiometric indicator. And what you do is you turn on the activity in a slice, and then you turn on the light once the cells are active, and then you have calcium influx, and this indicator is sensitive to calcium. So what you get is the ratio between green and red will tell you the amount of activity in the slice. And the nice thing about this experiment particularly, it was done virtually like overnight experiment from injection of the virus to the experiment. That's about 16 hours worth of time. And the reason is because in this particular experiment, we used ultra-fast viruses. Ultra-fast virus specifically, and I'll show you when I say fast, what do I mean fast? All right, so this is what you see here is a time-lapse imaging of a culture, mouse culture that express GFP using this virus. And what you see here, right, is not days, it's hours. Right? So I'll start it again, and you can appreciate how fast this thing is happening. I just have a look. After you get like 4, 5, 6 hours, and you start to see overall expression, which is fantastic. And so we wanted to make use of that in order to, to work with our human tissue. So the virus that we're using is the semeliquiforous virus, which is RNA virus. And it was initially found in Uganda, was isolated from mosquitoes in 1942. It's a disease pathogen, and essentially it's an RNA virus, part of the alpha virus family. And its carrying capacity is quite good, so we kind of can work with it to majority of or most of the things that we want to work with it, right? It's about up to 8 kilobase, and it's bigger than AAV, so that is the advantage. Of course, it expresses really fast, right? That's a major issue. And how do we get the viruses? That's, from my perspective, this is pretty simple. We go to our viral core facility and we ask them, can you please produce us a semeliquiforous virus with a particular construct? And they do that. It took time. It wasn't easy at the beginning. It was a kind of learning curve. And they developed the method of working or generating these RNA viruses. And the thing which is unique about this virus is this thing here. So this is the genome of the virus, and it has this promoter, the subgenomic promoter, which is very potent. So together with the RNA-dependent RNA polymerase, it kind of make copies of the RNA instantly, and a lot of them. So it's not only the fact that you get RNA into the cell, right? It's also the fact that you have very strong promoter, and it essentially duplicate the RNA pretty quickly, and you get this expression. So here is a first experiment. It's in rodents, right? And we want to check that it's indeed functional. So what we get, essentially, the products are actually functional. So what we did is in this case, it's one photon experiment. So we inject the virus. That, again, that's a kind of a overnight experiment, which I think some of the students, kind of a dream, right? You just go in, you inject, tomorrow you come and do your experiments. And you can see that depending on the number of spikes, you're going to get a change in fluorescence, and it's pretty comparable to what you get with AAV after about 2 weeks, right? And so we did similar experiments with channel rhodopsin just to see that things are functional. And indeed, you get function, but you can notice that the reliability, if you look, so we injected the virus, and then we patch one of the cells, we turn on the light, right? And then there are all these spikes that we generated by light pulses. And some of the cells fire more, some of the cells fire less. And here we start to see that it's not like the AAV injection when you have the maturation of the channels and it goes to the membrane, everything's done. It's pretty quick, so you don't get as much as fidelity, but if you can see, the first spikes are pretty good. So if you're patient in this, you get the first spike, it's going to work for you. Now, it's not a new virus, as I told you. And essentially, it became a viral vector for the use in a lab back in 1991. And that was striking for us because we thought, why didn't anyone use that? But people have tried to use that before. And one of the things is that you can see that immediately, that it's obvious that the expression level is very fast, right? So why people haven't been using that, and that is the reason. The curve that you see here basically tells you that the stronger, the faster the expression, the more toxic is the virus, right? That was like the main problem. So kind of people stayed away from this kind of virus, right? So it's like almost inherent problem of the virus. But remember, that's an old virus, and we have today so many new methods in order to control the molecular biology of the virus. So I'll just show you that if we only care about this duration of experiment as 24 hours windows, right, that is only what we care about, then it doesn't really matter if the cells die afterwards, right? If we care about long-term expression, that's probably shouldn't be for us. But if we care about a short time window, why not, right? And so we checked the viability many, many ways in order to get a handle of how toxic that's going to be within 24 hours. And this is one of the tests that we did, showing you that the number of dead cells in a culture is going to go up in these particular conditions. It's going to be indifferent to whether we injected the virus or not. Later on, so if you wait for 4 days, that's going to be fatal for the cells. But if you're going to work within the window, then it's fine. And so checking also on activity, right? So you want to see that the cells after, in this case, 24 hours or 22 hours are actually functional. And you can see in this movie that the cells will generate a calcium activity if you put high potassium and some gabazine. They will generate activity. And even if you look at multiple, if you look at the cells specifically, right? So these are all these kind of ROIs of the different cells in the culture, and these are 3 different wells of multiple cells. You can see that they are all synchronized. And so what you have is that the synaptic machinery is in place and everything is working within this time window. So we have to check the physiology, and I'm not going to tell you, I'm not going to stay too much on that, and the physiology is good, cells are healthy. Again, within this time window, but it's not 100%, so we have a little bit of differences, and that happens in the resting potential of the neurons. There is a little bit of elevation, and then we have the threshold is also elevated together with that. Right? So we have a little bit of change, which we have to account to, but the rest of the things are fine. Okay, so now the relevance to here, which you probably already have in mind, right? So using viruses was done in the past. Several laboratories have tried that, but this was consistently done in cultures, and there is good reason for that, right? Because you have to wait. The question is, and we cannot do in vivo experiments. Well, just to say that if it is for the benefit of the patient, we might want to do that, but as for our little games, maybe it's not. And so the question was whether we can do this experiment by injecting directly into a slice. And the nice thing, so if you worked with human tissue, you know that when you get a slice, it could be 50% bad slice. And then you find an island of wonderful, cells which are very healthy, and then you want to work on that particular one for as many hours as you can. And that's a benefit. So if we go to these places and we infect these regions, right? So we have good access to good tissue. And we know that the cells are viable, cells in human tissue are viable for a long time. 48 hours if they're kept in good conditions, maybe less if they, just you put them on the shelf and you wait. So we have the time, we have this time window that we can work with. And I'm going to show you now the first experiment that ever worked for us in human tissue, because that was really a bumpy road. Initially, nothing worked, of course. The virus was bad, the tissue was bad, everything was not working. That's the first time it was working. And so what we are doing essentially is we go to a human patient with, the surgery happens, and we take the slices, we take the chunk of tissue, we slice it. We do the visually guided viral injections. Right, so we inject directly into the slice where we keep it for about 17 hours because it's a little bit longer in human. The time for expression is a little bit longer in human than in rodents. And then after the 17 hours, we start the experiment. Right? And that's the first time that we get an indicator. But we wanted to see specific, once we had these indicators, we were pretty sure that the calcium imaging is going to work because cytosolic, if you want, the cytosolic proteins are kind of easy to work with. But harder than that, that's going to be the membrane protein, right? So because we want to manipulate the circuit. So we first started with channel rhodopsin, right? And the expression was nice, right? We saw expression. By the way, one point just to understand about this virus is that it's very potent. So you can have one particle infect a cell and just light it up. It will make the cell express. You just need one particle. And that is amazing because with AAVs, you can have hundreds to make it work out. But this, you need just one. That's good enough. And so, as I said, we did a channel rhodopsin experiment. That was a big disappointment. Because despite the strong expression, what we get is that the voltage, what we get as a voltage was about 1 millivolt. And that wasn't so appealing because what would you do with that, right? And then we said maybe, so the total conductance of a single channel of channel adopts is pretty small, right? So what shall we do? Just we take a bigger channel and we'll express that. And we were lucky enough to have the Chrome Q, right? The Chrome Q that was developed at the Boyden lab. And we used that as a comparison to the channel adoption. It's quite big, right? So we thought maybe we'll do that, and that's going to work. And surprisingly enough, we got the same results. Expression was beautiful. We get a nice expression everywhere, but still the voltage deflection for a light pulse was about 1 millivolt. That wasn't so effective. We said, well, it might be that we don't need something bigger, we need something different. And we thought, if we have other channels that have different evolutionary paths, and we make use of that. So what we went to is to the carmine, which is the origin is from bacterial rhodopsin, and it behaves like originally it's a pump, but it is converted to being a channel. So we said it's very sensitive, right? It has big currents, and it's different. And what we see when we put that in a mouse, right? We see that after expression, overnight expression with Semmelweis forest virus, it gets us very strong firing. So you can see that if the light duration is high, and it also can be very precise to a very small duration of light. That is working pretty well with the mouse. And by the way, this is being exploited in the lab because that's starting to become easy. You inject, you come in the morning, you do your experiments. And now with the human, right? That's why we're here. And with the human case, it was also working, and that was our big surprise. I don't forget the Christian doing the experiment shouting, "It's working, it's working." And the thing is that, you turn on the light, and what you see here is a human slice, and you see an infection. So the red ones are the infected cells. These two cells here are the cells that are being patched. One of them was patched. The other one is one of the experiments. And you put an electrode here, and you activate. This is the schema of the experiment. And then you activate the cell with light, and you get to see the spike, so the cells can fire. What we get is around, it's not 100% of the cells are firing. We have about 70%, which is, for many purposes, it's not bad. It can be better, and we are working on getting this better. And you can see here, once cell fired, you get very high fidelity for the first spike, and then there is a little bit of reduction. By the way, that's not out of the ordinary for opsin, so that happens. And the last is a demonstration of what you can do with this, like the idea of circuit mapping is the appealing point. As I said, what you want is to do the same experiments as you do with mice or rats or rodents, right, in other animals, particularly in our case in human. So here in number one, essentially, there is this cell, and it is patched, right? And now you start to turn on the light on different spots, and you can see the effect of short and longer range connectivity that activate the postsynaptic cell in this example site. So here you look at location 1, and here you look at location 4. So you stimulate location 4, and then you record in the same place. You can see that there is this circuit, if you want, Semmelweis-aided circuit mapping that you can do in human fresh slices. I want just to stress that there are several ways to do these injection protocols, and some of them are pretty amazing. Some of them involve in just scooping out the brain and injecting in the brain, particularly that would be of mice, because in humans we want to see the slice. You're just injecting in the exposed brain, you just wait a couple of hours, you start your experiment, and then within 3 hours, you're going to start to see some expression. And same way you can chop off the head of the animal, do the injection, therefore you kind of change the type of experiment from one protocol to another, right? So you can do the entire experiment on a dead animal, and then you also see the expression. And of course, finally, the human experiments. So I'd like to wrap up here and tell you the pros and the cons, because kind of, you can think about it as version 0.1. Oh, we said 10 minutes, now you tell me 1. Okay. So, pros and cons, that's going to be the last slide. First of all, it's going to be strong and sparse expression, of course, depending on the amount of virus that you inject. It's S1, so safety level 1. That means basically we do the experiment under the injection is done under the in the electrophysiological rig that's going to be later on used to do the patching. You use the same stuff is used to inject on. Or you just take the pipette, you trim it a little bit, it's bigger, so you can inject the virus. The length of experiment, of course, the length of time for expression is short. We'll give all of you the rapid experimental turnover. We use every day almost the effect of the Campari in order to identify region of activity in vivo animals. That's kind of animal welfare, if you're interested, and to some degree you should. That's also useful. You can express it in young animals, and finally, you can work with other species if you don't have the chance to inject to them in vivo. There are some cons of that, and before you will ask me, because that is a very important question, whether you have specificity, you can have specificity of this expression. So right now we don't have. The specificity that we get can be either from the place of injection, you can be really precise at that, or you can even block some synaptic connections, so you can isolate excitation inhibition. But other than that, we currently don't have this specificity, but we are working on it, both in rodents and in human tissue, which is different things. We're working on it, some positive signs, I will tell. There was a fast slide maybe that you didn't see right in the result. Membrane targeting, it's not every construct that's going to work for you. You have to choose the right ones, and once there is a community around this, it's kind of easy to get. You just need the alpha users, right, to be courageous enough to do it. And then cells survive for beyond 24 hours. This is something that we're working on with some success, and so we can make that better than what I showed you currently. And then SFE production currently is difficult. Also, this is something that we're working, making it accessible as kind of production to everyone. So finally, I want to say thank you very much to these 3 students, to Christian Ebner and Zina Dominiak, who actually did a lot of work that was fruitless for a long time, believing that that's going to work. And then to Jakob Müller, who basically took it on first and started this entire thing, was pushing it. So Thorsten does beautiful magic with the molecular biology in the viral core facility. And many thanks to all of you and to the taxpayer who is willing to pay us to do this kind of job. So thank you very much.
Great. So I think we have time for 1 or 2 questions.
Just, it's exciting work, but the fact that you can get a single virus initiating the expression, you can track the rate of growth of fluorescence, and that will tell you about the turnover of the virus. The rate of expression after a single injection with a low titer will give you some indication about the state of the cell, and that will presumably plateau out just before it dies. So you might be able to get a window more of saying, under these states, the expression of viruses are within the normal range.
That's definitely a good idea. The thing is that the virus has expressed several things within the cell. It's not only the construct which causes essentially. So one of the things is to track the expression level. That's absolutely true. Yeah. Yeah.
And you can also do that for a lot of long-term inactivation, desensitization of channel activities, calcium transients. Recovery from that transient is important as well.
Right.
And
right.
Phasic stimulation or activation could extract
Thanks.
Yeah, thanks. Some of the other virus types which express quickly, things like SynBis, for example, have not been sort of taken up because of the immune response that they elicit so much. Have you looked at all at whether you get that kind of response from this?
Yeah. Semlic, so in the living animals, Semlic is considered to be immunogenic, so it generates this immune response to some degree. We didn't see that only once in all our experiments. There was one case where we saw this, and that was probably because there was, during the injection, there was rupture of one of the blood vessels, so that kind of generate that. And it was very clear, the immune response there was very clear. But other than that, we didn't see it in the time window that we are talking about. There are studies, as you're right, there are studies that were looking at that, and they report that this usually happens after 24 hours.